My name is Jonathan Rolland, and I'm coming from the CNRS in Toulouse, in France. And I want to present you today a project that started 4 years ago when I was a postdoc in Rus Hoelzel's lab in UBC in Canada. And it's about the genomics of lateral adaptation in Chinook salmon. So lately I've been particularly interested in the adaptation to latitude, especially the genes that show a lateral decline in frequency, and identifying those genes may help to understand the current distribution of species, especially because those genotypes might be likely on the move in the coming decades. So we've been particularly interested on the largest species of the Pacific salmon, which is the Chinook salmon. And it has a distribution between Alaska and California. And it has a very complex life cycle from the river to the sea. So the eggs hatch in the river, the larvae and the smaller salmon goes down the river and then grow in the sea during several years and come back to the exact same river to reproduce. And what is particularly amazing in Chinook and generally in salmon is that we also breed them, and so we have some good candidate genes for timing and maturation. So why do those particular traits are important for studying latitude? There is a migration timing, spawning timing variation that relates with latitude. So you can see the red dots here, and Chinook salmon actually at high latitude enter the river to spawn earlier, so in June and July, than the fish at low latitude, December and January. And so one of the first candidate gene I could find was clock gene that basically has a polyQ domain, so a polyglutamine repeat domain inside the gene. This particular gene was associated to the day length at spawning and latitude in Chinook and chum salmon. And it was found in the sister species of Chinook salmon on an inversion that show a very strong variation in frequency with latitude. Another amazing study about the timing of migration is this intergenic region between rock-1 and grebe-1L. So it has been shown to be strongly associated with the spring run and the fall run of Chinook salmon. So in California, you have those two ecotypes in the same river. And they actually found that this particular region, intergenic region between rock-1 and grebe-1L, was implicated in the variation in terms of, so there was a strong difference in allele frequency between those two types of population. What is amazing is that the homozygous either migrate very early in the season or very late in the season, and the heterozygous have intermediate timing. And so about maturation rate, the Chinook salmon spend a longer time at sea, at high latitude, so they spend more years at sea. So 3 years on average, up to 8 years in Alaska, and 2 years on average in California. And this might be because the growing season is actually shorter at high latitude. Individuals may take longer to mature. And so there are two very important genes that were found to be related to maturation rate in salmonids, VGLL3, so vestigial-like 3, and Sino-Oculis-related homeobox 66. So VGLL3 is an adiposity regulator that is explaining nearly 40% of the variation of age at maturity in Atlantic salmon, while 66 is associated with the age at maturity in sockeye and steelhead and on the run timing of Atlantic salmon. So we sampled, thanks to a wide network of collaborators, the whole lateral range of the Chinook salmon. I really want to thank today all these collaborators that sent samples to us. Dolph Schroeter was the coordinator of that. And we went through, so we sequenced 137 Chinook salmon, whole genome, in 29 populations across the whole distribution. And so we went through a pipeline where we sequenced with Illumina, so short reads, aligned on reference genome, called the SNP with a truth and a training set, and we got nearly 8 million of SNP. So it took me 10 seconds to say and 4 years to do. So just two objectives of the analysis. We wanted to test the 5 candidate genes that I just described and their relationship to latitude. And then we had a more discovery phase of the genome to try to fish for genes that were potentially important. So because we didn't want to rely on a threshold that could be pretty arbitrary in terms of genome scan, we decided to rank all regions and to pick the top 5% of all regions as a threshold. It means that intergenic and genic regions, we had like 70,000 regions total, and this corresponds to 3,500 regions. And so we had two different methods, either BGLMER, that is a generalized linear mixed effect model with considering river as a random factor, and BayPass, that is a linear model. So always like you look at the occurrence of the SNP and the latitude, and you account also for the population structure. So we had two ways of summarizing the information from those models that we computed for every single position in the genome, every single SNP. And we measured what was the average p-value for the region or a 50 SNP window. So I'm going to show the result. So it's pretty small. The most important is the bold part of it. We found that all of the candidate genes had at least one of the methods that were falling in the top 5%. CLOC1B and CLOC1A were found to be significantly associated. Oh, this is too bad. Okay, so it's not showing up. All right, so let's imagine few SNPs in red here. So those are the top 5%. And actually here you have, for each of the SNP, you have a variation in allele frequency. And so you just have to imagine it's very terrible. There is actually a strong relationship in frequency between latitude and the frequency of each of the SNP we see in CLOC1A and CLOC1B. I hope that the other one will show up. For Grab1L and ROC1, we actually found that, again, here there is a huge peak. There is a huge peak of SNPs in the intergenic region that is actually, so this is not too bad. It is actually this peak in the intergenic regions that we can see. And it is matching roughly what Thompson showed in the winter run, spring run difference. But it's not exactly the same SNP. So there is something there that it can be pretty interesting. There were 56 SNPs that were in common, but what we show is that our region of strong association, and it's probably the regulatory region of ROC1, it is actually closer to ROC1 than to Grab1. And for VGR3 and 66, okay, I'll let you imagine it's very much associated. Okay, so for the discovery phase, I went through a wide literature review, and it was pretty interesting, but it also can last for years, right? And so related to clock, so we were looking for other genes that might be potentially related to the candidate genes. And so the clock feedback loops is very much is well understood in terms of circadian rhythm. So I tested all the genes in this circadian rhythm. And we also found pride, that is cryptochrome, and pride-hash and pride-1, and reverb that are inside this transcription feedback loops to be associated to latitude. VGL3 has been proposed to be related to the ICO pathway that is implicated in cell proliferation and organ size. I also tested those genes that were described in Turco, and we found a lot of the top 100 regions. So it's not like the top 5%, it's really like the top 0.01% that are also falling in this pathway. So I cannot go through all the details of all the genes that I tested in this different pathway, but what I can tell you is that it is mostly following a hypothalamus-pituitary gonadal axis that has been already described in many other fish physiological pathway. So basically going from the eye, taking the photoperiod, analyzing, getting the photoperiod in the pituitary, that is basically the orchestra of the hormones in the body, and having an impact on the gonadal hormones in the life cycle. So I think it's pretty interesting to go further. So the take-home message is that the genes related to the timing of migration, clock and ROC1 and GRB1R, and the maturation are likely key for the adaptation to latitude, and a larger number of connected genes may be implicated, but they may follow preferentially some physiological pathway. For example, the hypothalamo-pituitary gonadal axis. Thank you very much. I want to thank all my collaborators, Dolph Schutter, Chris Christensen, and more generally the University of Victoria for the help on the genomic analysis. Thank you very much. Yeah.
So you focus on the candidate genes and do the genome scan or the association studies. Do they show up, or do you need to look with a magnifying lens to find those associations?
You mean on the whole Manhattan plot? There are like hundreds of regions associated with latitude. So that's why I didn't show it, because there are basically peaks all over the place. And this is also mainly because like there might be also some population structure going on across the lateral gradient. But yeah, so that's why you have to go through and ask. But I think that in terms of p-value and false discovery, given that we had relatively few candidate genes at the beginning, I have good confidence that we found some relationship. All right, thank you.
I want to first start by thanking my many collaborators who are listed on the bottom here. You'll see their names pop up throughout the talk. Some of them are probably maybe even surprised to find their name here, but if they're in the audience, I hope I do you justice. So this is a large collaboration between my lab at Louisiana State University. I'm Prosanta Chakrabarty. We did the phylogenomics stuff on this NSF Rules of Life grant. Brooke Fleming and her postdoc at the time, Callie Crawford, who's now at Coastal Carolina University, did a lot of the biomechanical stuff that you'll see that's published as well. And Larry Page did a lot of the CT scanning work that you will see as well. And this is a story of convergence between lobed finfishes, which we belong to, the Sarcopterygians, and a ray finfish that wants to be a tetrapod. So it's a convergence of 400 million years. And we start with Tiktaalik. So this is the holotype of Tiktaalik. It's about yay big. And it's about 2 hours from here at the Canadian Museum of Nature, which is just on the Quebec-Ontario border. You're in the province of Quebec, if you didn't know that. And there are 9-foot specimens of these guys, but this is the holotype. And if you don't know about Tiktaalik, there are GIFs of it walking onto land. There are beautiful figures of it. There are funny memes about it. We wouldn't have the problems of the world today if we had shooed Tiktaalik back into the water. We wouldn't have humans to mess up the earth, etc. But going back to the future for a minute, this is a ballotoured loach. You probably haven't seen a fish perch. This is a perching fish. They live in Southeast Asia in fast-flowing streams, and they do something most fishes don't do, which is hang on to rocks and to things like this. And this is a strange behavior that we want to know more about how the anatomy is related to that. And so this is a CT scan of one of the extreme examples of a ballotaurid morphology. In purple here, you will see the connection between the pelvic girdle and the vertebral column. That's a hip. Fishes don't have hips. These fishes do. My lovely assistant, Christian Mack, who's an LSU postbac, will pass around a CT scan of this if you want to see in person. It's not a real skeleton, so be careful. But this morphology is quite unusual, and so we want to know how that relates to function. And so this is one of my proudest PI moments. This was my student at the time, Pam Hart, who became a biologist because she just learned about Cryptotara. And this is me bringing her to the one cave where it exists, and she's looking very proudly at them, at the fishes that inspired her to become one of the best cave biologists in the world. She's at University of Alabama recruiting students. And this is our Thai collaborators in the cave. Did I hear a roll tide? What happened? And so we spent 7 hours in this cave. People were eating, people were smoking. That's what that smoke is. It was a very unusual experience. But it was a fun cave, and this is the creature itself. So this is Cryptotara. It's a great swimmer. It's blind, doesn't have any pigment. It can go vertical on a cave wall, up a waterfall. So they're called cave angels. They're amazing little creatures. They are both creepy and horrifying outside of the water, however. So this is it walking. This is not a fish, it's a salamander. It's walking with the salamander gait. It moves its hips. Fish can't do that because they don't have hips. These do. And they even raise their head, which is unusual because it doesn't have a neck. So Tiktaalik does that. So we want to know more about it, and thinking about some cautionary tales. So this very great paper from the axial skeleton of Tiktaalik came out just this year, and it also has expanded sacral ribs going through the pelvic plate. And we want to know, what does that tell us about anatomy? We don't have videos of Tiktaalik walking. So what could it do? How much can we say about it? To their credit, the authors of that paper don't say much about the behavior. They say the anatomy of the hip evolved before forelimbs and digits, etc., in the aquatic environment. And I also wondered, if we didn't have video of Cryptotara, what would that skeleton tell us about function? And it gave me this, I always remember this quote, structure without function is a corpse, function without structure is a ghost. How much can you relate to function from anatomy alone? If those of you know Peter Wainwright at UC Davis, that's actually his father's quote. So we compared the anatomy first with, let's say, a regular fish, like a goldfish. They have uniform ribs, no connection with the pelvic girdle. Meanwhile, in ballotauras, these hill stream loaches, there's 3 different morphotypes of the rib, the expanded rib as it goes through the pelvic plate, the basiopterygium, with ligaments, etc., attached. And we were curious if these anatomical differences were variable enough to be statistically different. They are. So they truly are 3 morphotypes. My lab did the first genomic level phylogeny of these. So this is ultra-conserved elements, about 1,000 of them. For the first phylogeny of the group, first we noticed the taxonomy is just awful. That's somebody else's problem for now. But the ribs, so Cryptotara with the expanded big ribs, their closest relatives actually have thin ribs. So the rib shape is not phylogenetically conserved, which we thought was interesting and a little bit puzzling. So Brooke and Callie, they took this landmark-based approach. We brought video equipment to Thailand. They set up shop with these video equipment that they put up very quickly. It was quite impressive to watch. This is the reduced phylogeny of these that of the observed in the wild members of Ballotauridae. And we just asked, how does the morphology impact the walking kinematics? Brooke and Callie are not taxonomists. They don't get to name species like me, but they created computer programs in MATLAB and R. And I love that 2 women came up with the program Thick to describe the gait and distance per step model that they created. That's the tail hip insertion. That's a very millennial joke. Someone can explain that to the others. So this is, all 4 of these have the deep, robust rib shape anatomy. And some of them would not walk. So Homolopteroides on the bottom right there would only swim. And so that told us the morphology doesn't always tell us about function. If we do that thick analysis, some are very steady walkers. The gait is sort of shown by the amount of the shape of these little lines here. And so what that told us, since these all have the same anatomy, but not the same function, that you need the anatomy to walk. But the anatomy itself doesn't necessarily make you a good walker, if that makes sense. So you need that anatomy, but doesn't necessarily make you a great walker. And morphology and habitat are not as good a predictor as relatedness, which was still a puzzling result. So I thought that was a cautionary tale when we're doing paleo. If we're just looking at anatomy, sometimes we don't have the creature to videotape. This is not an XROM video of Cryptotarra walking. This is a sort of a nature fake of two undergrads basically wobbling the skeleton. You can't do that, to actually see how these things walk anatomically. You can't do that with Tiktaalik either. And to their credit, no one has done that, right? But some people extend how much the anatomy can go into function. Now, the story doesn't end there. We did the genomics. So this is collaboration with Brant Faircloth, who's my LSU colleague who's here. And the first thing we noticed that the genome is very small. So it's 560 million base pairs. Most Teleos, Actinopterygii, and rayfin fishes are about 1 billion base pairs. And I just had an undergrad do a survey of cave fishes because I thought maybe it's small because it's cave fish, doesn't have eyes, doesn't have pigment. But there's no real rhyme or reason with genome size for anything, but even within cave fishes. So in black are the cave fishes, and in blue are their sighted relatives in the same family. And sometimes they're right in the middle, like Astyanax. Sometimes they're smaller. Sometimes they're bigger. It's not, the genome size doesn't seem to be related to those loss of those elements in cave fishes. So this is going to be another cautionary tale. So I'm going to show you 2 slides where we compare the Cryptotarra with the stone loach, which is in a different family. And then I'm going to show you an update of these slides in a minute. So first we notice there's hundreds of duplications of the transcripts, these functional elements of the genes, and thousands of missing transcripts, just between Cryptotarra uniquely. We noticed opsin genes that were missing, but I've described cave fishes with no eyes at all with functional opsins. But these had some missing opsins and some duplications. The color gene was missing, which makes sense. They don't have any pigment. A number of clock genes are missing too. So there are 3 of these missing, and this makes sense. They live in complete darkness. They don't know light from dark. They weren't even aware of our presence. They're clueless about day and night. Hox genes were missing. We looked specifically at Hox genes because we wanted to know if this hip morphology was related to a Hox mutation. But the Hox genes that were missing were related to eye and pigment. There's duplication of a developmental gene and an insulin gene, which is important for some cave fishes and related to metabolism. But again, these are all comparisons with a sort of distant relative. And this cautionary tale is like whenever you see a genome comparison of mouse, chicken, human, or distant relatives, I didn't think about that much until we did this. So we, just in the last few weeks, got the genome of a second ballotaurid species, Homoloptera. The genome is tinier than Cryptotarra. In fact, it's only 50 million base pairs bigger than the smallest known vertebrate genome of Takifugu, the Chinese pufferfish. So it's incredibly small. And so, again, not related to cavefish size of the genome. Now, order of magnitude, less duplications and missing transcripts. So the story has changed quite a bit because we have more data from another ballotaurid comparison. So it seems that the opsin genes are lost at the base of Ballatouridae, not specifically to Cryptotarra like we had thought previously. And so these opsin genes are not the story that we thought they would be. The color gene, MC1R, is missing only in Cryptotarra, which again makes sense. It doesn't have pigment. But the clock genes, only one of them is missing now. But this is good. This helps us narrow down the story of which gene, which transcript is most important for circadian rhythm. And so that tells us a better story. And on the bottom is the string analysis, which is much more humble than what I was telling people a month ago, right? And so that's just another cautionary tale about when you do genomic comparisons, genomes are cheap enough now that it's probably worth putting one or two other close relatives into your mix. So to conclude, Cryptotora is a hip fish. If you haven't seen the model going around, please get it and make sure it comes back to me. But it has convergence, this ray fin fish convergent with the lobe fin fish, like us. And morphology doesn't always predict function, so the hip morphology might not be the most important pre-adaptation. Again, it's probably digits and limbs that allowed fishes to come onto land and give rise to us. And Cryptotora's genome reveals notable duplications and deletions, but fewer when you have a close comparison. So 2 cautionary tales there. And I'll thank you for coming. Thank you. Corey.
So you mentioned that they didn't notice you were there. Do they not have any predators or competitors?
It was really fun. So thanks for mentioning that, Corey. So there was another cavefish in the same cave. They didn't seem to overlap, but there was a pool here and a waterfall that had the concentrated area of where Cryptotora was. And maybe where the end of the room was another species of blind cavefish. And they're just eating other blind animals, invertebrates. But these guys, they crawl on land, so they move around. So it was very strange to me that they didn't overlap in the cave.
I wondered if you saw any changes in like genes related to behavior or neurogenesis or something, since they seem to not have as many other species interactions.
Yeah, I'm not sure how many of the transcripts for that are known from the stone loach. I think that's a little bit beyond what we can do right now. Maybe compared to zebrafish or something, but fish behavior, I mean, like, what are they doing? I don't know. Yeah, Max.
So you say like it doesn't match with walking, but what about climbing? You know, is that a different locomotion than walking, and like the ones that don't walk
Yeah,
climb?
it's really this gait, this wiggle that they can do, because they have huge pectoral fins and pelvic fins that help them climb as well, and then they could grab. So it's not that it's not related to walking. It allows them to walk, but it doesn't necessarily allow them to walk, if that makes sense. Thank you.
So I'm Rus Hoelzel from the Durham University in the northern parts of England. I'm going to talk to you today about cryptic diversity in the deep sea, especially that driven by natural selection. But before I start, I just want to acknowledge the people who actually did the work, including postdocs Anders, Michelle, and George, and intern Fatih, but with many other people as well who are listed at the bottom of the slide there. This is a tale of 2 fish. Neither of them walk, but cool fish all the same. The orange roughy is the first one. Samples came from places with open circles on the map there, and I've circled in green Faraday because I'm going to come back to that, out on the mid-Atlantic ridge. These guys breed on banks and seamounts and along the coastal margins at about 500 to 1,500 meters deep. They don't mature until they're like 30 years old, and they lived up to 150 years, so pretty unusual fish. The potential for larval drift is, compared to other pelagic species, relatively low in the species. Anders and co. looked at over 1,000 fish from around the world at about 5,000 SNPs, and found that in ocean regions they were segregating pretty differently genomically. We focused on the North Atlantic, and there the distance between sampling sites was genetically relatively low. FST values were very small. Correlation on a simple isolation by distance was not significant. There's something there, but it's not significant. But if you instead look at the correlation with respect to habitat range, so only looking at the habitat in which they live, 500 to 1,500 meters, then the correlation was much stronger, so isolation by habitat. So then we looked for outliers putatively associated with selection using these 4 different methods here. And we looked at a couple of ordination methods, FCA and DAPC, FCA on the top, DAPC on the bottom. The outliers are on the left there, and the neutral loci are on the right. South Africa and Namibia, just for comparison. North Atlantic is always on the right there. The main point here is that for the outlier loci, Faraday seamount, which is one of two seamounts on the mid-Atlantic ridge we sampled, stood out very much compared to the others, which were all sort of on top of each other. So this is very much a work in progress. We decided, inspired by this, that we should do whole genome sequencing. So we did 42 genomes from 4 populations. And the initial results are consistent with what we expected. All the comparisons with Faraday show a large number of outliers. For the comparison I've shown here, it's maybe too many outliers, so we're still working on that. But for the comparisons against populations that are not including Faraday, there's very few outliers. So there was isolation by distance, but only along the habitat range. And there was selection on the mid-Atlantic ridge at Faraday seamount, not Sedlow seamount, for reasons that we have no idea at the moment, but we're working on that, and that's something for the future. So the next thing I want to talk about is differentiation with respect to the vertical environment in the sea. Horizontal is what we always think about, but if you think about it, in the deep sea, there's tremendous variation with respect to environment as you go into deeper water. So one bar is 15.5 pounds per square inch, and that increases by a bar every 10 meters. So at 1,800 meters, you've got like 2,600 psi, which is a lot of pressure. Ecosystems change because the prey community and the predator community also changes. You lose light, water gets colder, etc. But if you think about it, there's the potential for a boundary there. So between the mesopelagic and the bathypelagic, that's where you lose light altogether. But in the northern hemisphere, where we were looking in mid-latitudes, it's also at around that point that you get a significant change in salinity and water temperature, and the oxygen minimum layer is right around there. So something like around 1,000 to 1,500 meters, there might be something happening with respect to a habitat boundary. So our next fish is the round-nosed grenadier. These guys live in benthic habitat on slopes, but remarkably over a great range of depths, so from about 180 meters all the way down to over 2 kilometers. And for everything I'm going to tell you next, it really matters if they have strong diurnal vertical migration. And there isn't any strong evidence for that. There's some evidence based on diet, but there's no evidence to suggest that they go over the entire range, certainly not by day. And we had some early genetic evidence that there was at least one locus under selection that was differentiated by depth. So this inspired us, and we generated a reference genome for the species using Illumina and PacBio and RNA-seq for the annotation at 120x. And then we did resequencing. So we did 60 genomes resequenced, 14 to 16 at each of 4 depths. And our objective here was to compare depth, not geography or temporal differences. So we sampled these over the same day or two from locations that were within about 20 kilometers of each other. So hopefully focusing just on differences associated with depth. And we look at neutral markers You see the ordination plot on the left there, there's really no pattern, but maybe the most telling is the plot at the bottom, which is FST. And in the upper diagonal, those are p-values. So there's no neutral structure between these depths at all. But if you look at outliers, they do segregate into different clusters. And it's maybe a bit easier to look at it based on these Manhattan plots. So when we do any comparison of depths that don't include 1,800 meters, the deepest depth, the plots look something like this. Maybe a few outliers, but there's nothing much going on there. But for every comparison against 1,800 meters, it looked like this. So there was lots of outliers and strong indication that there's really something going on there across the genome. And so for those 370 odd outliers, we did a gene ontology assessment and got a lot of terms, but over half of them had to do with development or morphogenesis. And then some of them were non-synonymous changes within genes. The main thing was that at 1,800 meters, for the sample we had on this occasion, they all showed a fixed genotype at that depth. For these various loci, you can see that rock 1 is again popping up here, but we're a long ways from salmon, and we don't think it has anything to do with migration. In fact, we think it has to do with development, which is a pleiotropic effect of that locus. So then when we look at linkage disequilibrium for neutral loci, which are in black compared to the outliers, which are there in blue, and especially the non-synonymous outliers, which are in red, you can see there's a significant elevation for the outliers, suggesting some kind of concerted evolution amongst them, even though they're not close physically in the genome. And then we wondered, okay, well, is this like a weird thing that's happening just off the Hebrides where we were doing the sampling? So we picked 4 of the loci, and we looked at this pattern across the entire range of samples that we'd collected in the North Atlantic. And what we found was that on the y-axis there, we've got the proportion of individuals with the deep water allele. For the juveniles, which tend to hang out at about 1,000 meters, it's sort of 50-50. But for everybody else across the geographic range, they're showing this pattern with the deep water allele most common in deep water. This is about catch data, just showing that the juveniles do indeed tend to be mostly in shallow water, but the adults can be either in shallow or deep water. And colleague Thomas Rechnier at Marine Scotland did this latent class model based on very extensive catch per unit effort data, and found that, yes, the juveniles are sticking around at about 1,000 meters, but the adults either go deep or they stay at about 1,000 meters. So now this analysis is a collaboration with Carlos Gaza and others. What we did is, intrigued by all this, we extended the sample size per depth to about 36 fish, and the number of depths to 8. And then the upper right there, what we're doing is we're looking at either side of 1,500 meters, S, shallow, D, deep, allele frequencies, and showing that in fact, for these depth-associated non-synonymous change SNPs, there's a strong effect, even when we expand our data set. And then we wanted to look at the pattern associated with age. So all these fish were aged, which is a real challenge for this particular fish, but we managed. Then we looked at control loci. We had 10 SNPs at 3 control loci, that's on the lower left, and there was no association at all. But for the loci associated with depth, they either increased with age class or decreased, depending on the locus, which is suggesting some kind of frequency dependence. And then we looked at various methods to look for long-term signals of balancing selection, HCA, NCD, Baller mix, beta score, and Tajima's D, which is what I'm showing on this slide here. And for Tajima's D in green, for every locus, all 8 of those associated with depth, there was a highly significant pattern. For the 3 control loci at the bottom there was no pattern. They also looked at feeding ecology based on stable isotopes, carbon and nitrogen. That showed a significant difference for the deep versus the shallow water. Also morphology, using the measures listed in the table on the right, and a random forest cluster analysis, we had 87% assignment accuracy. So my take-homes here are that the cryptic diversity here is apparently associated with selection, either local adaptation, like in the orange roughy, or bouncing selections in the round-nosed grenadier. There was no apparent neutral differentiation, or very little, for the orange roughy in the North Atlantic, but a strong difference at these apparently adaptive loci, and suggests that our need to look at this sort of thing if we're going to conserve adaptive potential. And for the round-nosed grenadier, it was kind of even stranger. It was like they were distributing according to their genotype. It's evidence that this is supported by balancing selection, and it means, of course, that if we're depleting either shallow or deep water fish differentially, that's going to affect their long-term survival, because we're depleting important information there. And this species, according to IUCN, is in fact endangered in the North Atlantic. And that's it. Thank you very much.